Welcome back, everyone. I am Dave Friedman. I am in the Department of Neurobiology at the University of Chicago. This has been just an outstanding conference so far. We've heard so many amazing examples of the interplay between AI and the sciences, and how AI is accelerating progress in fields from molecular biology, chemistry, climate, physics. And now I'm excited for this session where we'll hear about how AI is being used to accelerate neuroscience. And neuroscience as a field has something special to offer AI in that, neuroscience has been inspiring approaches in AI for decades, given that the brain is energy efficient, can learn from few examples, and I hope we'll hear some examples of this in today's talks. So I'll start by introducing our first speaker, Richard Andersen. Richard is the James G. Boswell Professor and the T&C Chen Brain-Machine Interface Centered Leadership Chair here at Caltech. I've been following Richard's work for decades. He was just a pioneer in understanding the brain mechanisms underlying spatial functions, particularly for guiding eye movements and reaching movements. And he was also quite an early pioneer in the field of brain-machine interfaces. And so we'll be hearing about this today. And Richard, take it away.
Thank you, David. And yeah, this has been a very interesting conference. And I'll be kind of now at the science end, not the AI end. And what we do is collaborate with AI people, as you'll see. So for the outline of the talk, I'll be talking about two types of technologies that are used for brain-machine interfaces. One is recording electrophysiological signals, and that's the most typical type. But I'll also be talking about using ultrasound for brain-machine interface, and that's very new, being pioneered by our group and a couple of others. So a brain-machine interface works by recording neurons from largely the cortex of the brain, feeding them to decoders that decode the meaning of the pattern of the neural activity, and then that can be used to control external devices. And these typically are robotic limbs or cursors on a computer screen. And we work with tetraplegic subjects, so they've had high-level lesions at the usually at the C3, C4, C5, C6, that's cervical levels, and that renders them tetraplegic, which means that they can become completely paralyzed below the lesion. So they have some shoulder movement, but not movement of the four limbs. And they also lose sensation of the body below the level of the lesion. So it's a very devastating neurological deficit. And usually what happens is it interrupts a loop. So if, say, you are thinking of reaching out and grasping something, and you can see it, it travels through the parts of the brain that analyze visual information, then goes to motor cortex, which projects down into the spinal cord. And you can see that a lesion there would break this loop, but people can still think about making movements. So we can use that, leverage that by recording activity of their movement plans from the cortex. And the type of device, the implant that we use. It's referred to as the Utah array, or more recently, a BlackRock array. And I think the takeaway is that it's extremely small. It's 4 by 4 millimeters. It has 96 active electrodes on it, typically records from 100 to 150 neurons. And this, say, is on a computer screen what the experimenter sees. And you can see these little action potentials that are generated. And those are the signals that we pick up and analyze. And this is basically the steps that you see with the brain-computer interface. So you have this amplification of all the channels from the array. We say in one trial, we have n number of neurons, and these are the action potentials, often referred to as threshold crossings. That's when the activity goes over a particular threshold, indicating a spike. And we bin those at around 30 milliseconds, and then produce, those generate features that are used in model fitting using machine learning. Actually, we were doing this before machine learning became such a buzzword, but looking back, that's what we've been doing. And then that model can be used to control these external devices. So these arrays are, in monkeys, they only lasted a year, but these monkeys were healthy, banging on cages, sticking their fingers up there in the arrays and getting infected. In the humans, we find they last between 5 and now we have one subject that's out for 10 years. So this is quite remarkable. However, during that time, there's a steady degradation in the recording. So this is from one subject, JJ, and this is his motor cortex array in 2019. You can see these are action potentials here that have been that are being shown on one of those displays. This is then in 2023, and you can see that the signal is greatly degraded. So we have a great reduction in the number of action potentials. However, that's not the only signal that's generated and picked up on these arrays. Another is referred to as local field potentials, and they come from averaging the synaptic activity from a bigger volume of the brain than does the individual spikes. And so you can see there's spikes here on this recording. This is time and voltage, and you can see this oscillation going on, and that oscillation is the local field. And that contains some interesting features that, and they last a lot longer than the spikes, again, because they're averaged from greater distances. So if you have some failure nearby, you still have a pretty robust local field potential signal. So the spikes are, I think most of you probably know, a rapid depolarization of the cell body that comes when a neuron generates an action potential. The local fields, as I mentioned, they can come from synaptic potentials that come from inputs, and they come local processing. Now, with Zita Amami, who's a professor here in electrical engineering, we being whoops, her and Benjamin from her lab developed, along with Tyson Aflalo and Jorge Gemez from our group a way of taking in all possible signals that we could get from the neural recordings and using those to generate features and training a model. And in this case, it's fairly simple compared to many of the presentations today, but it's a one-dimensional convolutional neural network. And it picks up, so a single channel will have 8 convolutional filters. They generate up to 8 features, and then these features are used to fit the model. And so then this shows in more detail what the model is like. There's a feature engineering module. Again, these are the filters, waveforms that are a result of the filters, and then they're further processed to generate the 8 features. And then that's taken from each electrode. So we, again, have a large number of electrodes. We actually, in subjects, plant now about 6 of these arrays. So we're recording from over 300 electrodes. And these feature engineering models are then fed to dimensionality reduction that is used to fit the linear model and then control external device. So this is an example of our subject, JJ. He's going to be controlling a cursor. And first, it's without VNet, without this feature extraction module. And you can see that as he does it, it's a little erratic. There's some errors. These are using the threshold crossings or action potentials only. And now this decoder B is actually the VNet. And you can see that as a result, so he's using his brain to control this cursor, and it makes it much smoother. And so this is great because it's been able to extend recordings at least 2 years more. And we're going on 3 at this point. So this just shows what you saw in the video with threshold crossings. The paths are very jittery. With VNet, they're nice and smooth. The angular error is much greater for threshold crossings than VNet, and the time to reach the target is also much faster. We've used this VNet for a practical application. So this is a collaboration with a neurotech company called BlackRock and with the Ford Motor Company. So it's at first, when the BlackRock people came to me about this idea, I thought, well, that sounds a little crazy. But from our point of view, it's nice that it's a demonstration in the natural world. So you have all this stuff going on while the subject is driving a car. And so the question is, can brain-machine interfaces operate in natural situations? Another aspect of it was that used bimanual control. So the thumb, the subject would imagine moving his thumb. It's, I think, taken after the controller for the Xbox. I don't know. I haven't played it yet. But it has a joystick. And the joystick in one direction controls speed, in the other turning. And that's in the right hand, imagine movements of the right hand. And when he imagines movements of the left hand, that's a brake. So it actually is controlling these 3 different dimensions. So this is the setup. are, I should mention that we did a lot of tests with simulators, and we could show actually JJ was faster than all the intact, 20 intact control subjects except one. They were equal. And that makes a bit of sense because you're going directly out of the brain, so you don't have that additional reaction time of going through the body. So I guess it would be applicable for race car drivers, maybe. So the signal is amplified, decoded, and also this simulation simulated with the simulator called Carla, which is common for the driving industry. And so JJ was able to have the same score, not significantly different, for turning, changing lanes, braking, not getting infractions like running red lights, not running into things, having collisions. So basically, we can conclude that, at least from the laboratory tests, that potentially JJ could drive a real car. And so the test for that is he's in California. We, as I mentioned, send a signal out, which is, again, the steering, accelerator, and braking. And that's sent to a Ford Mustang Mach-E, and that's located in Michigan. And so that's done over the internet. And so it sends a command signal to the car. Then there's video feedback that comes so JJ can then drive. And the one drawback with this, we actually have now a contract to put him in the car, so it should be much faster. Right now, it's 200 milliseconds to get the command signal over the internet, 400 to provide the feedback. So what you'll see is a speeded up tape. He's able to drive at 5 miles an hour, but we hope here in California with the car here and him in the car, it'll be much faster. So this is an example. This Ford has what's called M-City, so it's a test track. This red is the acceleration. The blue is the steering. And JJ is here in his home. And it's kind of interesting. There are a couple of people in the car, one safety driver and also an engineer. But I think it's cool that you can see that he's steering with his brain. The safety driver is just there to make sure he doesn't crash their Mustang. Yeah, as you can see, I'm progressing here around the track. And so what I should emphasize is when we started this project, already the arrays had degraded, but we were able to complete it using this FENet, this AI front end. So this goes out for a while, I think. But let's see, he'll come to a tunnel soon. Yeah, but hopefully you're convinced he can drive a car. Okay, so the second interface uses functional ultrasound. And functional ultrasound has become a very popular imaging technology. It was developed by Mikhail Tanter in Paris a few years back, and it follows, much like fMRI, changes in metabolism that cause increase in blood flow within the brain. But in this case, instead of measuring the bold signal, the oxygenation on the hemoglobin, it's measuring the power Doppler. So it's looking at the Doppler signal from the ultrasound that comes from the movement of the red blood cells into the area. And so it's only about 12 years old. The inspiration that Mikhail had was to use very high rate planar waves. So they'll generate several hundred images, compound those images, getting the echo signals coming back, and then they make one Doppler image. And it requires a lot of computation. This is done in 2D, so that's nice, but we're working toward making it also 3D. And we're able to, we being Iconius, and which is the company that Mikhail Tanter has been able to get these frame rates up to 2 hertz, which was sufficient for us to do a brain-machine interface. Now, what's an advantage of this technology? One is it has a very wide field of view. So this is the interparietal sulcus, a sulcus that many of us neurophysiologists love. It has interesting properties within it, cortical fields, and it can go centimeters deep as well as wide. So you can see a lot of territory. And we've referred to it as being mesoscale. So if we look at spatial coverage versus spatial resolution, single neurons are really superior. But then you have these non-invasive technologies, and they have very low spatial resolution, like EEG is up to several centimeters. Whereas FOOS has very high spatial resolution, up to 100 microns, or even pushed to 10 microns. And very large spatial coverage. So it occupies this region between the non-invasive techniques and the invasive recording techniques. Now, it's minimally invasive, and what that means is that if you were going to have this as a brain-machine interface, the sound waves won't go through the skull. So you need a sonolucent window implanted within the skull. But this is a much more minor operation than actually putting electrodes into the brain, where you have to go resect the dura and punch it into the cortex. So that's an advantage it has as well. So the question was, could we then use this for a brain-machine interface? And to be able to do that, you need to, usually the images that you see are averaged over many Doppler images. But in our case, we have to, in real time, be able to use single trials to decode what the subject wants to do. And as I mentioned, the images have to be collected at a high enough rate to capture the changes in the image. So we did this first with monkeys, and so we had a window. We could take images, pass it to a LDA decoder after principal components reduction, then have the task. While it's performing, we'd send back the images that we're getting to further train our decoder. And the first thing we did was a very simple task. So it was a saccade task. So the monkey fixates a dot. Then a dot appears at a location of the visual field, and after a delay, he moves to that location to get a drop of juice. And after 100 trials of him actually making eye movements, we switched to real-time brain control. And in that case, he's just sitting there and thinking about making an eye movement. And interestingly, after he learns about this transition, so after a few eye movements, he realizes he doesn't want to work hard to get all that juice. So he learns that he can just sit there and think about making saccades and get a reward. This is showing the decoding. So this would be on day 22. There's the training period here. As soon as it gets above, excuse me, chance, it goes to brain control. And this is decoding 8 different directions of saccadic eye movements. So here you see the performance is not tremendous, although we've been able to do things since then to push it up as high as 60, 70%. And this is on then another day using the data set from this day. So that's on day 54. And what's so nice about this is it's stable. So you can see it takes almost no training to begin getting very good performance. And that's because you're taking a whole image and not a single neuron. And say in our recording arrays by day 84, it changes the cells change on the arrays. So each day with the electrical recordings, we have to spend a few minutes recalibrating, whereas this is a highly stable way to train the network. You train it once, and then you just kind of fine-tune it over a period of time. And we've taken this out for months, so it's very stable and robust. So with humans, we've you probably know that people have decompressive craniotomies when they've had traumatic brain injury. This is because the brain swells. The pressure is very bad for the brain, so they have a craniotomy. And so this is work with Claire Rubat and also Charles Liu at USC. So Charles has a subject here where he replaced the skull with a sonolucent plastic. And so you can see here is the actual surgery. And so this doesn't offer much of an impedance to the sound waves going through. And so here's our subject, J.H. He's playing a guitar here. This is a view of his vascular anatomy. So these are areas. This is the post-central sulcus here. And you can see these are 3 different ROIs. One is in a hot spot, one's not so much. And you can see very easily, you could be your own decoder, that when he's strumming and playing the guitar, he gets high activation. When he's at rest, he doesn't. Now, that's when, for us, the AI comes in, and this is in collaboration with the NEMA's lab and using neural operators. Lydia Lin is a MD, PhD student in our lab. She has a poster this afternoon, so be sure, if you want more detail, to go visit her poster. And also Bahare, who's a postdoc in Neema's lab. And in this training pipeline, which is shown here, is we pick a Doppler image as a ground truth, and and another image that it goes into the decoder. The ground truth, it takes is compared to the output of the neural network. That generates a error signal that then back propagates through the network, and neural operators in a multi-layer network it is used to decode. And the results are that they can get a 10 to 15% of the amount of time, sampling rate, or time per image. So that's a many-fold increase. And it's comparable. The behavioral decoding is similar, and it generalizes across mice, monkeys, and humans. And it's this greater computer efficiency will be really important now when we go to 3D imaging. So in summary, the better decoders using AI can extend the lifetime of these implants. The new, less invasive functional ultrasound BMIs can use these AI to improve the time in collecting frames. And we hope to continue applying these to more complex real-time natural environments. So these are the real heroes of the research subjects. They participate in this research without getting any overt therapy or benefit. They do it for the next generation of patients. And I showed pictures of people that were involved in the various studies. This are their current lab members, funding organizations. We have collaborations with many laboratories, science laboratories, as well as the medical schools and rehabilitation centers here in the area. So thanks.
Thank you. I was curious about why you were thinking of controlling a car with JJ using a joystick instead of a steering wheel. Is it harder to make someone control a wider, bigger muscle like a bicep versus a thumb?
Yeah, we could, because we do decode reach movements. So conceivably, we could have him imagine a steering wheel. I think we chose the Xbox because he loves to play video games. And in fact, he uses them between sessions. We have video games that we've put together that he can control and play. So it seemed like the easiest way to go at the time.